Great. It's another really wonderful honor to be able to introduce our next Welch Awardee, Professor Stuart Schreiber. Professor Schreiber is the Morris Loeb Professor of Professor of Chemistry at Harvard University. He is the co-founding core member at the Broad Institute, and now he is also the founder of Arena BioWorks. And he started his career, his Ph.D. work with R.B. Woodward and Yoshito Kishi at Harvard University, trained in the absolute best labs of that synthetic chemistry, started his own lab at Yale University in chemistry, where he continued that pioneering work, creating unparalleled, still studied syntheses of natural products. When he moved his lab to Harvard University, from Yale to Harvard, he really began to turn his attention to biology, and I think this really benefited all of us because his attention to both fundamental aspects of biology and the use of his skills in natural product chemistry really opened up entirely new fields. One of my particular really valued contributions that he made was done by his then graduate student, Jack Taunton, who spoke earlier, when he was studying the natural product tropoxin. And in that work, they identified the target histone deacetylase, which at that time really was the founding member of the epigenetic regulators, that enzyme class we now call erasers. So that really can be traced back to the whole of epigenetics really was launched by that target ID. Beyond epigenetics, his work, which I think he'll touch on in the lecture, of the discovery of the mechanism of action of rapamycin, FK506, and calcineurin have been pioneering both in the fundamental biology. For example, rapamycin is still, to my mind, the one true molecule that across all organisms has shown to be a life extension molecule, which is still mind-blowing. So that identification of mTOR as that central regulator between metabolism and hormone growth signaling, it sits at the hub of what I'm sure we still have decades to understand. So in each of these cases, I think somehow through a gift that Stuart has, he's been able to pinpoint important beginnings and continue to mine them for many, many more discoveries to come. And the molecular glues and molecules that he'll tell us are just continuing to give really foundational new medicines. So it's my pleasure to introduce Stuart.
Thank you, Kevin. Good afternoon, everyone. It's just a great pleasure to be here. I'm very obviously grateful to the Welch Foundation for these very happy circumstances. So happy to be here with my friend Pete Schultz, and we had a chance to share some thoughts on that topic last night. And very grateful for all of you. I'm really pleased to be invited by Kevin. This has been an amazing conference. You've done a really good job, and obviously so have the speakers. I think last night I mentioned the word humbling because some of the things I saw, and it was just added on today. But I think I hope the one thing we all take away from it is this seems like the glory years of therapeutic discovery. I mean, what you're seeing is not normal, right? The types of the new modalities, the new ways of thinking about medicine is a radical change in the way we think. So again, for the young generation who likes to think about having an impact on medicine, boy, thank your parents. They brought you into the world at exactly the right time. All right, so what I want to do today is I want to use this as an opportunity to tell you a little bit about the origins of my lab's work in chemical inducers of proximity. And I want to start and take you back to the late 1980s, where the mysteries of signal transduction were just becoming to be illuminated. So although the details were still unknown, the role of protein-induced proximity was becoming very evident. So by signal transduction, I mean, how is it that an extracellular protein never enters the cell, interacts on the surface, and yet it influences very specific intracellular events all the way into the nucleus, where depending on the extracellular factor, very specific genes are turned on or off. So by protein-induced proximity, I highlight some examples. So that extracellular growth factor may bind its receptor, but 2 copies of the receptor. And if the receptor's intracellular tail has tyrosine kinase activity, in broad and close proximity, you increase the rate of transphosphorylation. There's all kinds of proteins that have domains that recognize the phosphorylated tyrosine. In fact, in Jack Taunton's talk about lipid kinases, lipid kinases have phosphotyrosine binding domains. What does that do? It recruits it in close proximity to the inner leaflet of the plasma membrane, where it encounters its substrate, increasing the rate of chemistry. This process of protein-induced proximity happens over and over as the signal is propagated into the nucleus. The same processes bring acetyltransferases and methyltransferases to specific histone tails, methylate and acetylate them. And then there in the nucleus, there are proteins that recognize these chromatin marks, and they are the transcriptional apparatus. And so in the genes that are nearby their docking site, then they become either activated or repressed. So is this foundation, plus the chance discovery of chemical-induced proximity, and then I'll add the recognition that I'll describe bifunctional compounds can be used to recruit neosubstrates to enzymes that led to a new way of thinking about modulating biology and medicine with therapeutic benefit, because it almost seems miraculous that we can now add chemicals and induce proximity and cause very specific chemistry to take place. I mean, Ben Cravat's talk just kind of blew my mind, that you add a chemical and all of a sudden, the editing of an RNA, a very specific RNA, right, is induced by that process. That's kind of like science fiction stuff in the past, but now this is the way we think about medicine. So I would like to share with you some of the new techniques that we're using to discover these 2 classes called molecular glues and bifunctional compounds, and I'm going to illustrate the various functional consequences of these induced protein-protein interactions. So the discovery that I alluded to was associated with these 2 structures, these 2 compounds. These are natural products. They have very little structural similarity, or virtually none. They were studied by a then postdoc in my lab, Jun Liu. And, we just thought these had interesting effects on immune cells. And if we could figure out their target, that might help us understand that signal transduction process. So the target of both of these compounds turned out to be the same one. It's a protein phosphatase called calcineurin. But the way these compounds inhibit calcineurin was very unexpected. So one, they don't bind the protein, the target themselves. They bind, we call presenter proteins. Cyclosporin binds cyclophilin. FK506 binds FKBP12. And only after they bind does the actual sort of bioactive substance emerge. It's a small molecule protein complex. And that binds cooperatively with high affinity, high specificity. Another unusual feature, if you look at the asterisk, you'll notice these compounds are not inhibiting the orthosteric site, as Ben would put it. That actually makes them substrate-specific inhibitors. They're not only highly specific and very potent, but you can imagine that small substrates still come into this enzyme active site and large ones do not. All kinds of features of these molecular glues that open up new possibilities. All right, a couple years later, a graduate student joined the lab named Eric Brown. We had known that rapamycin was behaving a very similar way. We didn't know if it were a glue, then what was it gluing together? Rapamycin binds that same presenter protein, FKBP12, yet when it binds, it directs it to a completely different protein. And Kayvon alluded to this. This was, at the time, a previously unknown protein. We named it mTOR. David Sabatini is a graduate student in Saul Snyder's lab. Same time, we published our papers back to back, made the same observation, and David's done just brilliant job illuminating this, what turned out to be a nutrient response signaling network, among many other labs. But so, you know, now we have these 3 molecular glues, and at this stage, the possibility exists that this is just a really rare phenomenon, and it takes either very complicated structures, like the 3 that I just showed you, or maybe it's, you know, a billion years of natural selection, and maybe we chemists will just not have access to this kind of special property. Boy, do we know that's wrong now. Ben and I have speculated that, you know, it may be more often the case than not, that when small molecules bind proteins, they change their interactome, they act as molecular. They're just everywhere. We just weren't looking for them. Now that we know to look for them, you find them routinely. Now, the first hint that this may not be natural selection at work was when then graduate student Steve Haggerty was carrying out very different studies, and he discovered a simple, very simple synthetic compound, not shown, called we called it synthetic stabilizer A. And what this molecule did was to take, in contrast to FKBP12 and mTOR that aren't normally interacting, it took normally dynamically interacting alpha and beta tubulin and clamped it shut, just the way the complex natural products Taxol, discodermalide, apotholone are able to do. But this was a literally one-step chemical synthesis, low molecular weight compound that was incredibly potent. And so that's what we called that a molecular clamp. Recently, Felix Hausch's lab published a really amazing, they synthesized a library of 1,000 compounds and screened them and discovered a completely synthetic rapamycin. It actually is a molecular glue for the same 2 proteins, FKBP12 and mTOR. Recently, Schreiber et al. Reported on a discovery of a new natural product that binds, once again, that same presenter protein, FKBP12. This has got us thinking like there's something kind of special about FKBP12 as a presenter protein. And the structure of it bound to its target is now CEP250, completely unrelated to calcineurin and mTOR. But look at what it's doing. The small molecule protein complex is binding cooperatively to an otherwise featureless coiled coil with high affinity and high specificity, even the region of the coiled coil. So this just reminds us that this molecular glue mechanism allows us to do all kinds of things that I think are difficult to do with the chemical alone. Okay, so now take a step back. I'll say that the way we think about this is that when small molecules bind dynamic proteins in cells, they change their biophysical properties. And we can think of the small molecule protein complex as a neoprotein. These altered biophysical features often change the interactome. And it's this change that leads to the many different functional consequences. Now, I already made allusion to the similarity of these glues to various post-translational modifications as recruiting proteins, but I want to give you a second concept, which I think is a really powerful one, which is that First, I'll remind you, one thing we learned from Jim Wells, we've learned a lot of things from Jim Wells, but one going back is the idea that small molecules don't bind proteins randomly. They bind in evolutionarily conserved regions that are usually involved in protein-protein interactions, and he and Tim Claxton in his lab called those hotspots. So small molecules bind these hotspots. Now, here's an interesting observation, and we've seen some examples of this in a number of talks. Missense mutations enshrined by natural selection, meaning the ones we know about because they have a functional consequence, if you analyze them, they do not appear randomly across the three-dimensional structure of proteins. They appear either in or near hotspots. Secondly, over and over again, the functional consequence of these selected missense mutations is to involved in recruiting a new protein. So they're involved in neoprotein interactions. So I find this very satisfying because you have a hotspot and you make a mutation, change the amino acid side chain, and you change the interactome. Well, if you go to the wild type or the native hotspot and you add a small molecule, it's kind of mimicking a new amino acid side chain. And so is it that surprising that they induce protein-protein interactions? I missed one point. I'm going to go back to this one. It's on the title that may have confused you. I put this title in because I live in a little bit complicated world these days where I'm constantly dealing with some of our sponsors who are from the tech world. And boy, have I found out the complexities of dealing with tech in biotech. So I came up with this idea that I'm going to share with you now as a way to bond to the tech world. So in the tech world, they love to talk about inflection points. And the poster child of an inflection point was when an Apple engineer wanted to create a new app called Find My iPhone. I think it's now called Find My. And so he had the great idea of adding a GPS chip to iPhone 4S and Find My iPhone. So now you have your GPS chip, and if you lose it, you can track it down. Well, multiple other groups, and then a tsunami of groups said, like people in the hoteling industry, people in the ride-sharing industry, they said, scrap everything we're doing. I've read about some of like Airbnb and Uber and Lyft. It's almost comical what they were trying to do, like figure out ways to get cabs to figure out where you are. All of a sudden they said, oh my God, look, everyone's walking around with a GPS chip. So Airbnb was born, Uber and Lyft, and I like to think that there's some parallels to the biotech world, and I'm going to give you an example of that in a moment. Okay, now I said this an analogy to missense mutation. So there are so many examples, and we've seen ones in this conference already. I'm just going to pick one of my favorites. This is from work from the Novartis group in Basel. They were interested in the paracaspase called MALT1. It's an immune cell signaling protein that talks to NF-kappa B. So first of all, the structure, it's two major domains, and they're kind of linked together by tryptophan 580. And I was really stimulated by Jeff Kelly's talk. I feel like there's a lot of, both the genetics I can tell you about and the chemical is very reminiscent of his kinetic stabilizers. I call them an intramolecular glue. I think we've heard other terms here. I think they're all synonyms. They're the same concept. All right, so they phenotypically screened and found a binder, and they solved the crystal structure, and here it is. And look what it did. It binds where the indole ring of tryptophan 580 binds. It causes it to swing away, and then amazingly, this triaza indole is binding not only the same site, but even the same orientation of the indole of tryptophan 580. Now that gave them the following idea. One of the reasons they were interested is that MALT1 is a gene that is subject to a Mendelian disorder where a missense mutation of tryptophan 580 to serine is causal for an immune dysfunction disease. And when they saw this, they said, well, maybe this is an opportunity. And so they got patient-derived cells lacking the indole, lacking this, and they added back their compound, and lo and behold, it was a corrector. It corrected. It works kind of in trans the way the indole is working in cis. Just amazing. And again, just see these examples over and over. Here's another one we've heard a couple of times. The idea that molecular glues can be useful as degraders. So to my knowledge, this was first recognized by Nen Zheng and coworkers. They were studying the very simple chemical, a plant hormone called auxin. And auxin binds the auxin receptor and then glues together an E3 ligase. And it ubiquitinates and degrades. That's the origin of molecular glue degraders. Now, next was work of Handa, who discovered the target of these compounds, lenalidomide or Revlimid, and thalidomide, as cereblon. And then my lab, working with Ben Ebert, Steve Carr, and Jay Bradner's lab, working with Bill Kalin, followed up on these results. And sure enough, these compounds are molecular glues. They recruit neosubstrates to cereblon. Cereblon is part of a multi-protein E3 ligase complex, and that allows, by induced proximity, the ubiquitination of neosubstrates. And then Ben mentioned Christina Wu's observation, which really links this notion again of both missense amino acid mimics and post-translational modification mimics. So she discovered this well-known old post-translational modification where unstable proteins undergo bond scission, leading to these imids that she noticed looked a lot like lenalidomide and thalidomide. And sure enough, proteins having this PTM are docked into cereblon, and they're working in cis. They get ubiquitinated and degraded, just the way in trans the glues are bringing in neosubstrates. So things just feel really coherent. Okay, so I could have picked a number of examples, but I'll pick this one of this like GPS inflection point. I thought we were going to hear from Kayvon. I guess as the organizer, you're not allowed to invite yourself. That's a shame because Kayvon, your work with KRAS is just so inspiring and so transformational in its thinking. And Kayvon obviously taught us how to target this really challenging target. And now that we know how to do it, not surprisingly, people are thinking about, are there other ways? So the company Revolution Medicines have reported on this compound as another way to inhibit KRAS. Originally what they did is what they did specifically is to repurpose the same presenter protein, cyclophilin. They knew there were several natural products that bind cyclophilin. They took some of them and then using structure-based design, as they've described it, they put in an electrophile and tried to design in the best contour for a molecular glue, and originally targeting to the G12C allele of KRAS. I think I understand that they now have variants of this that don't even require the cysteine, they're pan-KRAS. But again, it's the molecular glue story. Intramolecular glues, and you can call them different things, but actually from this paper, I'm quoting their words where they call this an intramolecular glue. But they teach us, like, how do you target a molecular machine? So the NLRP3 inflammasome is a good example. It doesn't have an enzymatic activity, but we know that it ratchets its way through several conformations in order to function. They discovered a really simple chemical, shown here, that, these are their words, is an inhibitor because it's acting as an intramolecular glue. It actually binds 4 distinct domains of this molecule and tightens it up and only allows that conformation now to be the low energy one, and that's how they inhibit NLRP3. Now, the granddaddy of them all, I'm a little biased, as someone mentioned I think yesterday as a co-founder of Vertex, but yesterday Ju Chen gave us her beautiful story on how Trikafta works, the Vertex drug that is used to correct a Mendelian disorder. Although there are many different disease alleles, there's a common one, as she taught us about delta 508, phenylalanine 508, and then the amazing ability of 3 distinct chemicals to bind 3 different hotspots, and amazingly, synergistically. So each binding increases the affinity of the next one. That's why you put 3 chemicals in a pill, and they work so well, but then functionally they're synergistic as well. And Ju-Chin, I don't know if you're still here, but thank you so much for helping us understand the remarkable properties of these compounds. Next, so things took a really exciting turn just a couple years after the original molecular glue observation. And for me personally, I like to call this my Camelot period, because it took us to a collaboration with Gerry Crabtree at Stanford, one of the most creative scientists I've ever met, and one of the most decent human beings I've ever met. And he's like a brother to me. But it also allowed me to get to know trainees in Gerry's lab, some very remarkable people. I don't know if David Spencer is still here, but I definitely want to shout out to David, who played a critical role in these bifunctional that I'm going to tell you about. So he actually first looked at this in the context of inducing association of the T-cell receptor and chemicals turning on the T-cell receptor. I won't tell you about that story, but so the idea was simple. I told you it was becoming clear that induced proximity was the key to controlling biological systems. But and there were so many different biological pathways and processes, so we're gonna have to discover a molecular glue for every single one of them. No, so we have a simple little workaround. A bifunctional compound loses the cooperativity of glues. To a first approximation, bifunctionals just bind independently here. You need 2 different binders, and you come up with a linker, you link them together, and you effectively do the same thing, induce protein-protein interactions. And then, so to even take it further, to really explore the degree to which we could control biology, we took the first several, maybe 3 different systems where there's a protein here, really good binder, and a functional linkage that allowed these 2 things to come together. And we then fused these 2 proteins onto all kinds of signaling molecules. And Gerry and I started a company called Area Gene Therapeutics. We made kits freely available to the academic community. 1,200 labs use these. There's over 2,000 papers that have been published showing the myriad functional consequences of inducing protein-protein interactions without having to discover the glue one at a time. David Spencer was the first with the T-cell receptor signaling. I'll show you one of the fast follow-on as another example of targeted protein degradation. So we were excited when Peter Halley, who's a good friend, came and was telling me about his studies on oncogenesis of the papillomavirus. He said, look at this, Stuart, it's amazing. The viral protein E6 is a protein glue. It's binding 2 host proteins, the E3 ligase UBE3A and P53, causing ubiquitination and proteasome-dependent degradation of P53. And this is an essential step in oncogenesis. So Peter gave us the constructs, and Martin Prushy, postdoc in the lab jointly with Gerry, fused FKBP onto onto the viral protein E6, and then another one onto various proteins of interest. And we had a simple little chemical we called FK1012 because it's amazing. Steve Diver, postdoc, did an olefin metathesis of FK506, makes a double bond. It abolishes calcineurin binding, and it's now a bifunctional compound, right? And so you don't have calcineurin emission, but you can induce this association. And sure enough, this led to the targeted degradation of proteins. Now, a little historical story for you. So Martin's baymate was Craig Crews. And Craig, of course, has gone on in his independent lab and shown over and over the idea of what he calls PROTACs, bifunctional compounds that bring 2 proteins together that are native proteins without these genetic fusions. This is some more work from David Spencer. This was the first example of targeted cell ablation, not protein, but cell ablation. So a transgenic mouse was made with a tissue-specific promoter, and we constructed an FKBP12 fusion of the signaling domain of the FAS receptor as a way to induce apoptosis. These animals were very unremarkable. They had a transgene expressed, but they didn't do anything. But when we added FK1012 to these animals, inducing association triggered the fast signaling and led to spatial and temporal control in vivo in an animal with a chemical. And to my knowledge, this is the first example of this being used in humans. I mentioned Gerry Crabtree started a company called Area Gene Therapeutics. David went off and started a company called Bellicum Pharmaceuticals, and they collaborated. And in this New England Journal of Medicine paper, they reported on leukemia patients that were given a bone marrow transplantation. But prior to that, the hematopoietic stem cells were transduced with the same FKBP FAST construct. And 5 of the patients developed graft versus host disease, each of them with a single dose of this compound, Remeducid, were rescued of their graft versus host disease. So the attacking the whole body grafted cells were eliminated. So, and then, as I said, with these kits and many, many labs, but a big shout out to Gerry Crabtree's lab. I mean, they really pushed this in so many interesting directions, especially gene expression, a long list of things that they've done in this list. I put down at the bottom optogenetics. This is another fun little story. So, Carl Dieseroff, of course, is the inventor of optogenetics. Carl's a very good friend of Gerry. They talk a lot, and Carl was when he was an undergraduate, was one of my students. And so what Carl recognized was that cryptochrome 2 is a protein that will become induced to associate with itself, but not with a chemical, but with a photon. This is the basis of optogenetics. So what they do is they fuse all kinds of signaling molecules onto cryptochrome 2 and shine a light and turn on all kinds of processes. All right, more recently, we've been doing all kinds of things. I just go through quickly. This is some work of, the next 2 examples are work of a physician scientist in my lab, Will Gibson, very creative scientist. He just left and joined the faculty at Harvard Medical School at Dana-Farber. Okay, so this is our example of transcriptional rewiring. I've mentioned a couple times Nathaniel and Gerry have done just really beautiful work, and I hope we'll hear about that. But here's how we did this. So this is a bifunctional compound. We took a cytoplasmically anchored transcription factor. We did actually several transcription factors with IRF1, and it's got a nuclear export sequence. So we toyed around with different proteins that have a nuclear localization sequence that would be dominant. And we found in this case BRD4. And so a bifunctional compound that simply binds these 2 allows the NLS to dominate, and you can see changing cellular localization. This is the protein upon adding, it's going into the nucleus. But the beauty of BRD4 is it's got a very strong transcriptional activation domain. It induces liquid-liquid condensates that concentrate the transcriptional machinery. And so amazingly, when we treat with this compound and do gene expression profiling, we turn on uniquely the IRF1 transcriptional program. So a chemical can now turn on the activation of a transcriptional factor. One other example from Will, this one is in bioRxiv, and it's in press in Nature Chemical Biology. So a lot of people have looked for synthetic lethality with mutant P53, the most common mutation in cancer, and no one has found it. But what Will recognized as a clinical oncologist, and knowing that his surgeon friends, when performing surgery on a P53 mutant, will stain for the levels of wild-type P53 or mutant P53, because it's such a good way to get the edge of the tumor for surgery. And the reason for that is that mutant P53 is massively upregulated. So the cancer is trying, futilely, but it's trying to compensate for the loss of transcriptional activity by massive upregulation. So the synthetic lethal of mutant P53 is mutant P53. And so what Will did here was he took a P53 mutant binder and he linked it to a non-specific toxin, and then showed that he could concentrate this in cells harboring mutant P53. So we'll see if that's maybe a way to get at the P53 targeting. So I want to finish up with some new ways that we've been pursuing to discover either molecular glues or bifunctional compounds, but in the latter, not having to find a binder, linker, just all in one step. And then even to screen for molecular glues, cooperative molecular glues, where we pre-select whichever presenter we want and whichever target we want. And so this was a very simple adaptation of DNA-encoded library screening. So it started with a postdoc named Jeremy Mason, and what Jeremy conceived, so this is a normal DEL experiment. You have your target protein on a magnetic bead, you incubate with a DNA-encoded library, you have these barcodes, you wash away the nonspecific binders. There's a ton of nonspecific binding of the DNA to the magnetic bead, and then you just PCR amplify and sequence, and you look for barcode enrichment. The field calls that binary enrichment, and that's how you find binders. Jeremy's idea is real simple. Let's do the same experiment, but let's add our pre-selected presenter protein. And then we measure what we call ternary enrichment, okay? The barcode enrichment of these conditions versus these conditions. And that led to all kinds of bifunctional compounds, but the beauty of it was we were not discovering a binder and then a linker. It was just in one fell swoop, out of a single library comes bifunctional compounds. And we published on this last year and showed functional consequences of these with a range of target proteins. But what I really want to emphasize here is work of a postdoc who joined after Jeremy, was really inspired by Jeremy's work, and Jeremy helped Xuan Liu a lot, adapt this in another way. You may look at the dates and say, what's going on there? That's the publication process of like, the earlier work was published later, but whatever. So what Xuan idea is so simple but incredibly powerful. Like molecular glues have this very powerful property of being cooperative binders. And so she said, well, why don't we analyze data differently? Why don't we look at the ratio of the ternary enrichment over the binary enrichment? And that's really getting at the cooperativity that requires both pieces together. And this has just worked amazingly. This is one of the examples of targeting a transcriptional regulator. This was a bifunctional compound right out of a screening collection. X-ray structure shows the bifunctional compound is making lots of protein-protein interactions. The linker itself is making all kinds of interactions. And then more recently, you notice FKBP12 kept coming up, and we think that it's probably a really good intracellular presenter protein. And this has been borne out by a graduate student, Gerry Tan, who is a joint student, or was a joint student with Romnick Xavier and working in my lab together. And he has been looking at all kinds of different target proteins, but using FKBP12 as a presenter, and we find over and over ways to discover, with Xuan's method, the molecular glues of FKBP12 presenters. And here's a couple of crystal structures, and you see the different proteins that are shown. And then, oh, I'm sorry, one other one I'll say. The subject of this paper, by the way, links this to human biology in the sense that Romnick had identified a risk allele in inflammatory bowel disease, and that involves a gene called ATG16L1. It's actually not shown here, but it's in the paper. It's the main subject of the paper. And working out the mechanism, it suggests that what you really want to do in Crohn's disease is to stabilize this protein. And so we thought, well, what if we just stabilize it by inducing a protein-protein interaction? That's exactly what Jurian was able to do. And in fact, potentiate autophagy. I think we heard another talk about the various reasons you might want to do that. And the last one is not from my lab. This is a full disclosure. This is work that I was given permission to present from Brian Safina, who is the CEO of a company called Magnet Biomedicine. And full disclosure, both Ben and I are founders of this company, Magnet Biomedicine. But what Magnet is doing is saying, is this technology robust for all kinds of therapeutic applications? And I alluded to this in the introduction, but the idea that you can have a systemic drug now through the molecular glue mechanism, will not engage its therapeutic target, because that target may be throughout the body in different tissues. But if you pick the presenter to be restricted to the diseased tissue, the systemic drug can only become active where you want it to be active, in the diseased tissue. And so in this particular example, the diseased tissue is the vasculature, the extracellular space. And using just exactly same technology, using these presenter ratios, a magnet found a molecule that targets interleukin 17A in a glue mechanism, bringing serum protein together with it. The compound doesn't target either of these alone, but all together you form this high affinity complex. So sort of thinking a little bit about small molecules inching their way towards therapeutic antibodies, but through the conditional molecular glue. So anyway, let me finish. Thank you for your patience here. I want a big shout out to three colleagues in particular. I mentioned Gerry Crabtree. Ben and I got together, I don't know, 10 or 12 years ago as really resonating with this idea of binder first or function first. And Ben, with his powerful chemoproteomics, we hope that maybe the DNA encoded library world could complement, and diversity-oriented synthesis could complement. And then I only gave one example, but Ramnik Xavier is a close friend and colleague, and a human biologist, human geneticist, and a physician scientist. So I thank all of these people, and I thank you for your attention and patience.